There's another question from Veronica Ning. The last one was from Louise Laro. Veronica asked, Dr. Jacobs, for the CRUISE model, does your group check longer time points like 1 month after crush? Do astrocytes become more normal?
Yes, I mean, the astrocytes after an optic nerve crush, 1 month after optic nerve crush, astrocytes become morphologically more normal. And we have looked at their gene expression profile, which also seems to kind of return to baseline. We see after optic nerve crush, we see the biggest changes 1 to 7 days after injury, but 3 weeks and 3 months after injury, it kind of returns to baseline.
Okay, so it looks like we have questions now for the panel and for other speakers. Here's one for Dr. Sun. From Dr. Liao. Do you think there's a general IOP for do you think there's a target IOP for glaucoma in general? And what about IOP targeting patients with glaucomatous pattern of visual field loss in optic disc drusen?
Okay, I'm sorry, say the last part again.
What about an IOP target for patients with glaucomatous patterns of field loss, visual field loss in optic disc drusen?
Yes. So in my mind, I think you should be individual based. I don't really prescribe to a just a like a broad recommendation for everybody. But that said, though, I certainly think that closer you are, if everything being normal, so like if there's no corneal thinning, there's no steroids in the patient not taking those. If everything being normal, I think the pressure, if we can get closest to teens, mid-teens, like 15 or closer to 10, will be even better. So if I was affected and I had to I had optic nerve drusens, I would probably want my eye pressure to be as close to 10 to 15 as possible, and without of course taking more medicines with side effects, of course.
We have a couple of questions for Dr. Hu. Yang, do you think the calcium imaging result reflects the risk of cell death eventually in the glaucoma model? I'm not quite sure what it's referring to, but I guess in the calcium imaging result, do you think it reflects the risk of cell death in your glaucoma models?
I don't know, actually. So what I can say is that we can only detect the signal from the surviving cells, right? So for the cell that already died, we don't know. But in terms of the subtypes, like there are some subtype is more vulnerable to glaucoma or crash injury, and there are some subtype of the RDCs that is more resilient to the injury.
There's another question from Eric Geyer. Was there any change in RDC functional identity under normal conditions over the same time frame without crush or ocular hypertension?
Yeah, we do follow up that. Even more than that, we look at the contralateral eye of the crash in the same animal, right? One eye is crushed and the other eye is normal. We don't see any functional conversion along the at least 2 weeks period of time. However, in the glaucoma one, it is very surprising that one eye with silicone oil injection to increase the ocular pressure, right? The other eye is we use as control, so-called control. However, the contralateral eye, even without the IOP elevation, without the silicone oil, the activity of the RDCs is actually changed. It looks like there are overactivation of the cells of the RDCs in terms of in response to light stimulation. We don't know the reason of that, but we guess that's somehow compensate to the glaucoma eye which is lost some function. So the contralateral eye seems kind of try to overcome, but it's just totally guess. We don't know why is that, but we do see, if in the same animal, one eye has glaucoma, the other one has functional change for sure. Yeah.
Turning back to astrocytes, Tatiana, there's a question which I believe comes from Dr. Liao about whether you can comment on the diurnal pattern of astrocytic activity and taking out the garbage, and how this may relate to optic nerve health and development of optic neuropathies.
This is a very difficult question. I think it's really interesting. Now, astrocytes are not professional phagocytes, let's say, so they may actually take a pretty long time until they have gotten rid of whatever they phagocytosed. Now, we don't know about the diurnal pattern. Unfortunately, this experiment would be very difficult to do because of the enormous heterogeneity that is present even in a normal nerve. So we would have to do lots of animals. However, this is an interesting question, and I would suspect that it does, but we have no data on it whatsoever, unfortunately.
Dr. Young-Hu, there is a question from our colleague, Dr. Sui Wang. She wants to know if calcium levels can be associated with other biological processes other than activation inhibition of neurons, especially under disease conditions.
So that's why
your imaging.
Yeah, there's a lot of calcium signaling pathways that involve in this that we totally aware of that. But the way we measure it is that we measure it, the difference before or after the light stimulation. So whatever we get, the result is from the light evoked activities. So based on that, we can really pretty confident that that's the activity of the cell. But you're right. So in terms of other calcium signaling that involved in this, we cannot really totally ignore that. But using other sensors, for example, voltage sensor, will be more confident in terms of that. Which is part of the continual plan that for us to testing voltage sensor in addition to calcium sensor.
If I can jump in, we're also looking at calcium signaling with regards to regulation of the perinuclear cyclic AMP compartment. And in fact, we have preliminary data that we'll be following up on whether there's in fact calcium fluxes that may be independent of action potentials and visual information transmission that in fact are regulating cyclic AMP that's involved in survival pathways. So I think this issue, and particularly in neurons, we're just starting to scratch the surface about whether calcium is all about the action potential and information transmission, or whether because of the structure of cells like retinal ganglion cells, whether or not there's in fact going to be calcium is being used for other functions, in particular, in this case, whether we're worried about cell survival. So I think there's a lot more to be learned.
For sure, yeah.
I don't see any more questions in the chat or the.
I had an earlier question for Dr. Hu. I was wondering about the fantastic calcium imaging array that he showed. I was curious if you think these alterations in the calcium waves, do you expect that to correspond to eventual axonopathy or apoptosis?
I guess the simple answer is I don't know. So we need to do more, because this is actually data analysis is quite kind of labor intensive, and we got this data almost 1 year ago, and we take 1 year to analyze those data. And on one way it's good because it's kind of high school. We can get about 1,500 RGCs from one retina, right? And we can follow up them along the time. So the one way to do that is that we follow all of these cells, and then until they die. So we can get a kind of better sense of how this predict the death. But in reality, it's too much work to do that until we have a automatic way. Because for now, for the single cell tracing, we still manually to do it, which is expensive, labor intensive. So until we had an automatic way, maybe using AI or machine learning, that we can do that automatically, then we will have a better answer for you, I guess. But that's the goal. I totally agree. That's if we can using that as a predictor of the cell degeneration or even recovery, right, after treatment. So that will be fantastic.
I'm happy to ask a question. Tatiana, by the way, thank you for joining us. It's a real pleasure, as always, to get to hear about your work, which is always so wonderful. And it's kind of a question for you and/or Yang Hu, which is, astrocytes, are well described to network with each other. There's been papers in the past, maybe not in the optic nerve head specifically, but certainly in the retina and the brain around calcium signaling in these astrocyte networks. And I just wonder whether you or others, either of you or others could expand on, I don't know, your thoughts about calcium imaging of the astrocytes in vivo over the time course of disease and injury.
At present, there is one method that would be suitable for that, that has been published 2 or 3 years ago by Nguyen and Quigley, who just cut the optic nerve, mounted on an 2-photon, and then they can image for a couple of hours. I'm not sure if it has been applied to calcium imaging yet, but that would be possible. And otherwise, it would be very hard to image in the actual optic nerve.
Yeah, I don't know a way that maybe the green lines that can access into the optic nerve region could be possible. Another way we're kind of testing is called fiber photometry, which is a lower resolution. So we cannot detect single one, but it's in general. But again, that's still for axons, not for the astrocyte surrounding the axon. But if there's a way to do that, that would be fantastic. That's really a cool question to ask, how the astrocyte in response to this, to axon, which one is the major driving reason of the degeneration. That'd be cool to answer. But for now, I don't have a clue how to do that.
I have a question for all the speakers. In optic disc drusen, one of the key features is that the pathology is only in the unmyelinated optic nerve anterior to the lamina cribrosa. So can you comment on the glial or axonal properties that seems different between the two? What is it about the unmyelinated astrocyte supported axons that is so hard? Like, for example, Tatiana, I don't know if you compare astrocyte in the unmyelinated versus the myelinated region.
We have studied the unmyelinated region more intently, but it seems to be when we look at morphological changes, most what happens is in the unmyelinated region and the myelination transition zone. In the prelamina, for instance, the astrocytes do not change that much, and I never see them very phagocytic. So I'm not sure. I'm hesitant to say they are of different types, but most of what we can observe goes on indeed in the glia lamina and the myelination transition zone. I would like to mention that unmyelinated axons need about, I think, 70 times as much energy to propagate their axon potential. Maybe that's one of the limiting factors here.
So Joyce, related to your question, actually, when I look at Tiziana, Dr. Jacob's presentation, which makes me thinking, you know, I know you are trying to generate ODD animal model, right? So let's say if the astrocyte is really responsible for clearing up the mitochondria releasing from the axons. So I'm sure Dr. Jacob have a way to get rid of all those astrocytes. And then without those astrocytes, where the mitochondria will go, it may aggregate surrounding the axon to have a model. You know, it's just a simple thought. I don't know if that is possible or not.
Yeah, it's a good thought. What's really striking is, at least in the end stage where human autopsy studies were done, the glial elements were clearly described as relatively unimpressive. So it's not lost or reactive, it's just there. And then there are all these extracellular calcified mitochondria bits actually just sitting around, not being clear. So, you know, we're working hard to try to identify the first genes for optic dystrusin. And so, you know, I think that will give us probably the best clues. But we definitely wonder about the role that glia plays, because it must play some important role in this location in terms of drusen formation.
Hmm.
There's another question. I assume this is from Louise Lauro. I assume this is more for Tatiana, but the question is, can clarity techniques be used to improve visualization of gliomorphological changes in the optic nerve bed?
I've never done it myself, but I have seen images that people were doing at, yes, I've seen it applied to axons.
Yeah, it seems like we should inject the mitotracker into the superior colliculus, do the clarity, and then do some pretty cool in vivo 2-photon calcium imaging.
I don't know if you're still going to see much calcium waves after putting the optic nerve through all that.
Yeah.
Okay.
Well, if there aren't any more questions, Dr. Liao, should we break for a few minutes before we go to the next session?
That sounds great. So yeah, thank you, everyone. The talks were fantastic. I learned a lot. And our next session will start at 12. This is the patient panel.